Good afternoon, everyone. We're going to go ahead and get started. Can you hear me okay? It's on. Okay, perfect. Well, welcome to the seminar series for the Great Plains Ideas CTR. One quick housekeeping item before I introduce Dr. Lepley. We're going to hold all the questions for the end. So if you have questions, we'll get to those at the end and hopefully have a nice discussion. So I'll tell you a little bit about Dr. Lindsey and then, or Dr. Lindsey, you know, it's Dr. Lepley, the two of us. And hand it over to her. So she's an associate professor in athletic training at the University of Michigan. And there she also directs the Comparative Orthopedic Rehabilitation Laboratory and is co-director of their Orthopedic Rehabilitation and Biomechanics Laboratory within their School of Kinesiology. In 2014, she earned her PhD in kinesiology from the University of Michigan and then completed a postdoc at the University of Kentucky in their Center for Muscle Biology. She then took her first faculty position at the University of Connecticut and now has been back at the University of Michigan since 2019. Her research program focuses on elucidating the mechanisms that regulate skeletal muscle strength, activation, and structure after traumatic joint injury to establish interventions that optimize muscle recovery. And you'll see throughout her presentation that her group utilizes a non-invasive animal injury model and human subjects research design to design, test, and translate these strategies into clinical practice. She's been continuously funded from the NIH since 2018, initially through a K01 award, and this was followed by R01 funding that was awarded in 2022. Other accomplishments include authoring more than 50 peer-reviewed publications and the 2022 Dr. Freddie and Mrs. Hilda Pang New Investigator Award from the National Athletic Training Association. So please welcome Dr. Lepley, and I'll hand it over to her.
Okay, you can clap. I'll be taking questions now. Thanks so much, Liz, for the introduction. It's been like so nice to be here. It's been a really wonderful visit that was thoughtfully designed. So thanks for all of you that have made that happen. I'm going to talk about treating sick muscles after ACL injury, a bench to bedside approach. So I'm going to set a couple of ground rules and talk about a couple of fundamental concepts that we're going to talk about in this lecture in the event that you don't roll out of bed thinking about these things. I really like muscle sarcomeres. And these are sort of the foundational building blocks of muscle. You can think of them sort of as the Legos of muscle. And as a reminder, for those of you that didn't sort of like wake up thinking about them, right? This is why muscle is sort of striated underneath a microscope. When these contract, muscle gets smaller in terms of length. And the reason I like sarcomeres so much is they're really predictive of function. So whether they're arranged in parallel or arranged end to end, it'll tell you if your muscle is built for force or if your muscle is built for velocity. The other thing is that like the translation of animal models into the clinic is very meaningful at the muscle level. Structure, proteins are incredibly well maintained across species, right? So don't let it be like a turn off that a lot of this stuff is going to be in a rat. At the muscle level, it maps on really well. So, you know, I think my child and dog, you know, at the muscle level and at sometimes the behavioral level are like practically the same, okay? And so what we've done in our lab to sort of take advantage of this translational nature of muscle is we've built a device that also sort of ups our ante. Okay, so traditionally, if people wanted to give a rat or a mouse an ACL injury, they chased them around the lab with a scalpel and they opened up the knee joint, okay? The challenge with that is that just opening up the knee joint, not even taking out the ACL, sort of confounds everything that you're going to see. So we created an injury device that looks like sort of what happens on the field. It's a closed joint injury. And this improves our translation, right? Because now we're sort of able to look at the natural biological response. So let me show you how this works. We've nicknamed it the terrorizer. So essentially what happens is there's a linear motor at the top and a load cell at the bottom, okay? Rats are anesthetized, they're put to sleep, okay? And at the top, what happens is we have a program that drives a linear motor down at a certain speed. At a certain point in time, the ACL is super stressed out and it ruptures, okay? The load cell detects that, so the linear motor doesn't keep going. Like, there's not a ton of job security in this. Like, I can hear it from across the room if it's successful. We wake them up after we've done a Lockman's, just like any athletic trainer would do on the field, right? We do just a little baby Lockman's on their knee joints. If we don't feel any contraindications, which is really rare, we just wake them up. We also have approval through, we've worked hard with our Ira Cook team that we do not prescribe any pain medication. So you're looking at like the natural response here to these knee joint injuries. And it's important to have some basic studies to sort of substantiate the things that we do in the clinic, right? For a very long time, clinicians have said, hey, you're walking poorly, that's probably gonna be bad for like your bone health, right? But data that actually sort of connected these paradigms wasn't necessarily available in the ACL literature. So just to that point, right, what we do is we do a lot of, basically have a rodent lab that mimics what happens in the clinic. We just shrunk everything down like Honey, I Shrunk the Kids, right? And so what we are able to do in our lab is we look at their motion capture, and we're able to look at sort of their knee joints and how they move at times. So this is a look at the sagittal plane. It's basically their knee flexion angle. These are fancy functional data analysis plots. Basically what they do is they don't just look at like one point in time to say, oh, their knee flexion angle is bad. They look at the magnitude and the temporal progression of things, right? Anytime something's shaded, it means that's statistically different than a control group. And these are male and female plots transposed on each other because, you know, we just want to make things really complicated. So essentially what you can see is day 7 and day 14 after ACL injury, both male and female injured rats walk differently than healthy controls, but the points and stance in which they walk differently is sort of like limited to narrow windows. And then at day 28 and day 56, like majority of stance is sort of changed, where they just have less knee flexion. Okay, what we did is we mapped this on this relationship of like having less knee flexion, less peak knee flexion, and the subchondral bone health. And the reason we chose that is we looked in the weight-bearing region of the bone, right, of a knee that's most closely associated with the articular cartilage, and we found that it was really sporadic in rats that didn't achieve this knee flexion range of motion, right? So biomechanics truly do matter to knee joint health after ACL injury. Okay, why do we choose to study ACL injuries? Well, they're really meaningful. There's a lot of them that happen. They're expensive. They happen to a younger, vulnerable population. And at this moment in time, surgery itself doesn't protect the knee joint against getting this post-traumatic osteoarthritis, right? We, in particular, think about saving the muscle to save the joint because this persistent muscle weakness, quadriceps muscle weakness, has widespread complications. It's linked with a reduced re-injury risk, depressed quality of life, reduced physical activity levels, and knee joint health. Right? So for us, like if we can improve this like one thing, like it sort of has far-reaching effects. I think the challenge for clinicians is when a patient walks into the room and they have sort of depressed knee volume, it's not super clear what factors are regulating that volume loss, right? And in the case of ACL injury, there's a lot of factors that are sort of like lurking underneath the surface that are causing muscle to sort of be challenging to rehabilitate. For perspective, like 1 or 2 of these factors in any other disease condition would be challenging to overcome. You have a deck sort of stacked against you, okay? And our lab motto is clinicians are never gonna get this treatment right unless they have an accurate understanding of disease and disease progression. They need a better roadmap, right? And we, as clinician scientists, need to provide that, right? So the trajectory for our patients is better. Okay. Neural compensations are very widespread after ACL injury. This is a really, well, when I was doing my dissertation work, this is a really pivotal paper that came out in 2010 that talked about factors at the periphery, at the spinal level, and supraspinal sources that are sort of contributing to this inability for somebody to contract their quad well after they've had an ACL injury. This was a strength and conditioning coach on the Michigan football team when Rich Rod was a coach, which may sort of date the time of this video, but it's still relevant. I think that this person as a strength and conditioning coach should know how to do like a quad contraction, right? Their livelihood is based on it. And so, you know, this is a person that's ACL deficient, so some of these pathways are probably in play. I've asked him to just push his knee into the back of the table, and you can see like his vastus lateralis definition, like he's pretty much able to cue it. Okay, same person, 24 hours later after patellar tendon graft surgery. I promise the video is playing, right? This is what a lot of patients look like when they come out of surgery. Now, this is probably bottom of the barrel, right? This is the first couple of contractions that this person's trying to do. It will sort of get better, but it's really challenging as a clinician if somebody can't even activate their muscle well to get them to exercise that muscle. I put this paper out with one of my favorite collaborators in the last couple of years, my husband. And we sort of updated that Rice and McNair paradigm based on how the literature has evolved in the last 10 years, where there's this understanding that after ACL injury, there's factors at the periphery that sort of dysregulate things at the spinal cord level that lead to changes in the way that the brain sort of reorganizes itself or uses different centers to activate the muscle after ACL injury. What I've done more independently in my lab is this area of investigation, sort of this final common pathway. What about the direct connection of the nervous system with a muscle? Like, how is it being maybe compromised there? And thankfully, we have a model in which we can explore that, right? So what we did in this experiment is we invited a bunch of rats into the lab, and then we tore their ACLs, okay? And then we sequentially put them down to look at the time events in which muscles sort of became sick or dysregulated after ACL injury. So sort of a highlight of these results is before any factors associated with muscle atrophy came online, what we saw was first sort of this neuromuscular stress response in rats at 6 hours after ACL injury comparative to healthy controls. What I'm showing you here is some immunofluorescence that's looking at NCAM. And if you're cool, you can say it like that at a party, but it stands for nuclear cell adhesion molecule, okay? When you're growing and developing, you have like a lot of NCAM on the outside of your muscle fibers, muscle cells, because it helps with like connection to the nervous system. It's kind of like a glue, if you will, okay? But during periods of injury, it also lights back up because it senses sort of this neuromuscular miscommunication, okay? And what you can see is after injury, it's very brilliant in our animal model. At later time points, you know, the typical machinery that comes online to sort of say trigger muscle breakdown was evident in this model. But I think the important thing is it's kind of like the lights are being turned out. There's sort of an electrical disconnect between the nervous system and the muscle, and maybe that's a really key thing that's sort of tripping things up. I have a PhD student now, here he is, Luke, who's sort of following this up in a more intensive way. So what he's doing is he's taking male and female long Evans rats, and we're surgically implanting EMG into their quadriceps muscle. The reason you do that is if you just stick something on the side, they will just eat it off, okay? So we have to surgically implant this so it's all sort of inside, okay? The rats recover from this surgery, and then what we do is we get their baseline sort of volitional muscle activation while they're walking, okay? And then we provide them with an ACL injury, and then we serially track these things over time where we're also looking at their gait. And what Luke is finding is that in male and female rats during these early time points between 6 hours and 7 days, you know, there's a lot of depressed muscle activity on the order of like 30 to nearly 40% when these rats are just using sort of a regular gait cycle. So this would sort of mimic what we see in the clinic in the sense that there's this reduction in volitional muscle activity. What Luke is doing now is he's looking to basically see, like, is any of the machinery broke, right? Is something on that final common pathway an issue? So he's looking at the health of the neuromuscular junctions, and he's also looking at the ability of the motor branch of the femoral nerve to send a synaptic connection to the muscle, right? We need to rule out if these things are involved or not involved because a lot of like therapy depends on sort of the structure of these devices. So Luke's, you know, digging through that data in the next year or so. I think we'll be having some papers that come out relative to that. I think independent of, I guess, what's happening at a higher level, the end result is that there's a loss of synaptic activity that's going to disrupt the postsynaptic environment, right? And it's a domino effect. There's a lot of postsynaptic processes that depend on electrical activity to regulate them. And those things are now at risk, right? And in our hands, and this is the beauty of a hallway conversation, we've been able to link other sort of critical elements of muscle that seem to be dysregulated after ACL injury. So when I was a faculty member at UConn, I had the good fortune of having a new faculty member join, Oh-Sung Kwon. And yeah, I was talking to him in the hallway, and I was like, hey, there's a lot of fiber type transitioning that's happening after ACL injury. People are becoming more type 2. Like, what do you think about that? He's like, have you ever looked at the muscle mitochondria? And I said, nope, but I'd love to, okay? And so what we did is we designed this study together where we looked at mitochondrial function after ACL injury in our preclinical model. And what we found was, in terms of function, is that they have really depressed and long-lasting issues with their ability to basically utilize oxygen well after ACL injury. They produce upwards of 200% more of reactive oxygen species, so it's excessive. It's associated with that fiber type switch that you see in humans. It becomes more type 2 and smaller. And this is like really simple. We did more fancy statistical modeling, but these events are essentially related with each other, okay? We followed that up with other sort of metrics of mitochondrial health. And we see that processes that are involved in sort of like the regulation and homeostasis of the mitochondrial environment are disrupted in our preclinical models. So that continues to substantiate this idea. And what's been really important is in the last year, people have shown that, yep, this happens in humans too, okay? So the same issue with the depression and sort of mitochondrial function is being observed in humans. What are other things that go sideways after ACL injury? We know there's a depression in satellite cells. These are kind of like the stem cells of muscle, so just generally having more of these around is good. We know that they're down after ACL injury. You know that's a more pro-inflammatory environment. Generally, that's not great, right? We also know that genes that regulate muscle growth are disrupted, namely myostatin. So myostatin's a negative regulator of muscle growth. This is what a regular whippet dog looks like. This is what a myostatin deficient whippet dog looks like. In the case of ACL injury, myostatin goes up, so they don't look deficient, they look even skinnier, okay? And we also know that the tissue becomes more fibrotic. So, you know, like when I tell my grad students, like when you go to a bar, you know, and you sit there and you order a burger, you want the size of your burger to be good, but you want the quality of the meat of your burger to be good too, right? And the quality of the meat is bad. It's fattier and more fibrotic. Okay, so I would contend that it's no wonder that recovery after ACL injury is so difficult because there's so many things that are lurking underneath the surface that are just giving people a hard time. I think traditionally, this has been viewed through sort of like an inhibition neural problem at a higher level. We're starting to think about this at a smaller scale. So this disruption of alpha motor neuron activity and its consequences on sort of the postsynaptic environment, how the postsynaptic environment depends on that, and in particular, mitochondria seem to get pretty tripped up, and how that production of excessive ROS could lead to contractile damage dysfunction and muscle atrophy and weakness. Make sense? Okay, I'm going to take a quick drink of water. I'm going to turn the page now and talk about good things associated with ACL injury paradigm. Okay, so I've been kind of doom and gloom, and I don't want you to leave here without thinking that there aren't some promising solutions to that. So let me talk about that now. This is a really nice paper that Grant Norte's group put together a couple of years ago, looking at, I guess, the cumulative nature of all the different types of therapies that people have tried after ACL injury to help with recovery of muscle function. And two that I really like a lot that I'm going to talk about for this lecture are the power of neuromuscular electrical stimulation and eccentric exercise. And the reason is that I think these therapies can help promote activity at the neuromuscular junction, and or they can mechanically trigger the system in a different way that helps promote muscle health. I'll also give a nod to this. I think the more that we understand about ACL injuries, the likelihood of there's some work being done here that pharmaceuticals are probably on the horizon, because this sort of depth of inability to recover muscle health is sort of necessitating different solutions. All right, so let's say you go the route of, I want to promote activity at the neuromuscular junction. I think NMES is a very logical choice for that, neuromuscular electrical stimulation. I think it's part of the standard of care. Most people use it after surgery, right? But I think the effectiveness of this is sort of like the Wild West. So this is a forest plot, although it doesn't look like a tree. And what I'm showing you here is like the effectiveness of high frequency NMES to restore strength after ACL injury in a bunch of different clinical trials. And what you can see is that the longer it's been around, maybe the more questionable its effectiveness has become, which is disappointing. Okay, the studies that blew it out of the water were in the 80s and 90s. Okay, We took a hard look at this, and part of the R01 that we have now is to sort of investigate, well, why, right? How do you deliver in a way to sort of maybe replicate these results, and can we replicate those results in our preclinical model? I think the clinical viewpoint right now, if you're going to use NMES, the clinical viewpoint is to give them a lot of it with a unit that can deliver a high intensity. Okay, there's a nice paper put out in the journal JOSPT in the last year that basically advises clinicians that you have to crack open the manual to make sure that the device can deliver a strong enough punch. Okay, you want as much area under the curve as possible. Okay. And so I think if you're going to go with high frequency NMES, this is the way to go. It's noteworthy that the 2 studies that blew it out of the water use this device, okay? The VersaStim Model 380. If you see one of them, call me, okay? These things deliver the electricity basically out of the wall, right? And so that's why in our mind that we think that these interventions were much more potent than anything else that's been done up until now, because maybe it was just a much higher and more intense intervention. Okay, I'm about to contradict myself, so I want people to like hold on to their desks. Okay, so I would say that this is the clinical viewpoint. I will tell you that when I talk to muscle physiologists about this idea of like, let's just jack the muscle up with as much electricity as possible, they would say, why would you do that? Okay, and it lies in this like dual role of reactive oxygen species. They're not always bad. Okay, so this is kind of a cool figure that was put together, oh gosh, 20 years ago now, that's looking at sort of the ability to regulate the amount of ROS that you have in your muscle, and that at some point there's sort of this homeostasis, right? So here's some basic explanation. If you're not exercising and you're using antioxidants, that's maybe not great for you, okay? If you have way too much ROS, which is probably this condition in our ACL injured rats, it's also not good for muscle function. Okay, there's sort of this sweet spot where you have this balance of free radicals against its own sort of endogenous defense. You with me? Okay. One of the ways that we use NMES right now might, and there's a kind of some basic science data to support this, is that you're like hammering the muscle with way too much electricity. And it's tipping this balance to where you're sort of creating more free radicals and you're sort of upsetting the scale, okay? So if you're using high frequency NMES, maybe the reason it's not being as effective as we think it is now is that it's too much for the muscle to maintain, okay? The attractive thing about this is you just need to, so you're kind of over here if you either don't use it or maybe use it at too high of a frequency. The attractive thing is that you just can toggle the frequency down. So in basic muscle biology studies, there's a lot of compelling evidence to show that low frequency NMES delivered at sort of for a longer time point restores sort of this balance and creates sort of this defense mechanism, okay, where you can kind of balance it out and move towards here. So as part of the R01 we're doing right now, we're looking at the standard of care. We're giving our rats as much e-stim as possible with clinical units that we just sort of MacGyver down, okay? But we're also looking to say, look, should we just augment some of these parameters and maybe it can help different components of muscle? Or maybe there's a time when you want to promote strength, so you should use high frequency NMES versus you need to promote an antioxidant defense, okay? I think we also need to provide clinicians with different solutions, however, because some people just don't like stim, right? And so there has to be other things that are available for them. And so now I'm going to change pages and talk about eccentric exercise, so the power of negative work. Sarcomeres, we're back at them now, right? For a very long time, people thought about muscle, the functional Lego unit of muscle is sort of being actin and myosin, your thin and thick filaments. And they didn't consider, because we didn't know about it, that there's this other huge protein running through it called titin, okay? And I really wish I had come up with this saying because it's very cute. Titin is the blueprint or molecular ruler for sarcomere architecture. So this is a sarcomere that I'm showing you up here. What's really cool in some basic science experiments is people have shown that if you elongate a muscle sarcomere, okay, so that means you have to do a lengthening contraction. What happens is that the center of the sarcomere at the M line opens up. There's a gated mechanism, okay? And when it opens up, it creates a binding site or sort of a trigger to say, grow more muscle sarcomeres, put more Legos in series here, right? Be protective to muscle. So if you lengthen your muscle, it's mechanically triggered to basically grow more. If you shorten your muscle, it cannot open up that gate. Okay, here's a different way of looking at it, right? Muscle, it can be stretch activated. So this kinase domain is what we're looking at sort of in the center of their muscle sarcomere here. With concentric exercise, tighten, this springy element is coiled up. Okay, but if you elongate a sarcomere, that spring is going to uncoil and it's going to stretch activate the muscle and send a signal to the body to basically say, grow more muscle tissue. Okay, so from our perspective, like, wow, this is at no cost and it's a really effective way to make muscle volume bigger. The challenge is there's a lot of fear associated with this type of exercise because it's commonly associated with muscle damage. So is that true and where did that come from? So let's do a brief history lesson. Eccentric contractions, why are they so odd, right? When a patient comes into the clinic, you basically have one of three options, right? You can do isometrically biased exercise, concentric, or eccentric, okay? The definition of eccentric, when you apply it to a person, like, oh, that person's a little eccentric, they're odd, right? We applied it to muscle contractions because we didn't know how they occur, right? So that's why they got this namesake, okay? And it wasn't until, well, Hugh Huxley and A.F. Huxley are really largely in part responsible for telling us how concentric and isometric contractions occur. So this is just like a cool tidbit. Hugh Huxley and A.F. Huxley, not related to each other, on different sides of the world in the 1950s, basically gave us the cross bridge theory. Okay, so how does muscle get shorter? Hugh Huxley observed it and A.F. Huxley mathematically proved it. Okay. But what A.F. Huxley, and they both got Nobel Prizes for this, so, you know. But what they didn't do, this would be an example, this is his mathematical model that if you're taking my neuromuscular class, you'd have to know. But basically what A.F. Huxley did is he proved mathematically how isometric and concentric contractions occur on this side of the scale, but he put a question mark for eccentric contractions and he sort of left it there. Like, I don't know, this is confusing, right? But he still got a Nobel Prize, okay? Where did this idea of damage in eccentrics come from? From Boston. There were 4 members of a gymnastics team that performed a finger extension flexion task, okay? And they did way too many of them, basically, okay? And then they came back into, this is the figure from that paper, they came back into the lab, 24, 48 hours later, and all said their fingers were sore, okay? And so this idea behind delayed onset muscle soreness and eccentric contractions boils down to this study, okay? That people get sore fingers from doing way too many finger lengthening task exercises, okay? But this really sent into motion this idea that this type of exercise is damaging. The other thing that happened is that people use basic benchtop models to better understand muscle damage, and those messages got a little misconstrued when they hit the clinic, okay? So what you're looking at now, this is a mouse. This mouse is under the plane of anesthesia. These electrodes are stimulating the muscle, right? Simulating a muscle contraction. And then what they've done here is they've chosen a muscle with a long tendon because they've looped that tendon to a torque transducer. So what this allows the scientists to do is pull the muscle, right, at certain lengths in a very controlled way to know, like, oh, if I pull it this much, how much torque does it produce versus this much, right? It's a controlled experimental design. The challenge is that I don't know very many people that run around with tendons disconnected from their bones. What's the major function of a tendon? Absorb strain, right? So these muscles were very vulnerable to strain injuries because the anatomy of the muscle was adjusted, essentially, right? So like in healthy muscle, right, this is what it looks like. It's multinucleated, but the nuclear content is not located inside of the muscle cells or muscle fibers. And then it got really scary, after people did these experiments and they looked at muscle health, between 3 and 14 days, right? There's a lot of muscle damage that's occurring here. But they were able to pull the muscle this far because the tendon was no longer connected to the bone. So they pulled it so much further than somebody can pull their muscle in the clinic, okay? And so it sort of established this idea between fiber injury and lengthening contractions, but it was misconstrued, right? Because they anatomically adjusted these animals to make this observation. The other things that have sort of substantiated this idea between damage and muscle injury. You know, there's these basic force-length relationships. A.V. Hill did a lot of phenomenal things for muscle mechanics, but he called the descending limb unstable. So basically, when your muscle gets long, I don't know how you get force enhancement, but the connection between actin and myosin is unstable. Dave Morgan in the 90s also came up with this idea of, oh, maybe you get more force from doing an eccentric contraction because every time you do it, you pop a sarcomere. And that like liberates force, okay? Think about it. Every time you walk downstairs, right, your muscle's damaging and liberating force, okay? And then there's just been like a ton of articles, right? A lot. So if a clinician were ever even considering using eccentric exercise, they would maybe go into PubMed and they would Google these terms, and it just wouldn't line up for them, okay? So our job is to sort of better understand, like, are these things true? And if they're not true, then let's put some work into the literature to sort of refute this dogma, okay? One of the things that I guess led people to these ideas and observations is that they just thought of muscle as the two-element model, actin and myosin. And it wasn't until they started adding titin in that it explained everything. The interesting thing about this is that in science, I would contend that we don't know a lot about most things because they're too small, okay? But titin, we didn't know it was there because it was too big, okay? So for those of you that have worked in sort of a basic muscle biology lab, one way in terms of like sort of quantifying muscle health is you look at protein content. And you look at protein content by doing a Western blot. And most Western blots are about a note card big, okay? And those allow you to look at actin and myosin, which were both 40 and 200 kilodaltons. They're pretty light in weight, okay? It wasn't until people started pouring Western blots that were the size of notebook paper that they were able to see that titin actually exists too, and that's 3,000 kilodaltons, okay? So once we understood that titin is there, it started to explain everything. Muscle is not just two elements, it's three elements, okay? And so conventionally, right, muscle is unstable when it's lengthened because it's just relying on these thin and thick filaments. But when you plug titin in, it starts to explain everything about these models. So we just kind of like missed it, okay? So what are we doing in our lab to help counteract this therapeutic belief? We're basically conducting experiments in animals in which we don't change their anatomy. The tendon's connected to the bone, okay, to prove that, hey, this is not associated with muscle damage. So a very simple experiment that we conducted a couple years ago is we ran a rat on a treadmill, many rats, okay? And we either ran them uphill or downhill. These rats have never seen a treadmill before. When rats are running uphill on a treadmill, they're doing concentric contractions with their quadriceps muscle. It's a muscle shortening action. This is not fancy. We just tipped the treadmill down, and now they're doing eccentric contractions, okay? And what we looked to see was like, hey, if they only run for like 15 minutes with 5-minute bouts and 2 minutes of rest, which I think is very reasonable, right? Do they actually have muscle damage? And so what we found is that in rats that engaged in just a single bout of eccentric exercise for 15 minutes, basically 24 hours later, is that they had a very robust protein synthesis response on a downstream marker of the mTOR pathway, okay? We also found that this was associated with like very minimal amounts of muscle damage. We looked at things like IgG, which is basically an element that lies on the outside of your muscle cells. It's too big to get into a cell unless it's lysed or ripped, okay? And when it's ripped, it can infiltrate and glow. And so this wasn't a concentric animal. We just basically saw normal muscle remodeling in these animals, okay? So no damage, right? Because we didn't adjust the anatomical system, right? And a pretty robust protein synthesis response. So how does eccentric exercise work? There's data to show that it helps with the proliferation of satellite cells. We know those are depressed after ACL injury, so it's a good idea to maybe have more of them. Maybe they produce more power because there's this sort of idea that it helps with selective recruitment of fast twitch fibers. I think that there's good evidence to show, and there was just a human paper that came out to say eccentric exercise results in sarcomere genesis, which is a fancy way to say more muscle Legos, okay? And then we've also shown in other contexts that it also has some neurological benefits in terms of improving their ability to use their volitional activation. What do we know right now in terms of prescribing eccentrics after ACL injury? Is that, wait for initial graft healing, usually 6 to 8 weeks. We have shown that 12 treatments are beneficial and long-lasting. So 2 treatments a week for 6 weeks, that's pretty easy. And that you at least need to train in a way that you're sort of overloading your muscle. So above 60% of your 1 rep max, okay? My collaborator, Ryan Palmieri Smith, is running an R01 clinical trial looking at this at a much larger scale. So I suspect that we'll be able to report more things to you in the years to come. I think if at all I've convinced you that this might be a good idea to think about using after ACL injury, I wanted to provide some resources. So here are 7 different papers that are using eccentric exercise, showing beneficial results with varying degrees of equipment and varying degrees of timelines after ACL injury. So this is sort of a cookbook, right? Take what you want from these different protocols and use what you think might be advantageous to your patients. That's all I have. Good? Great.
Okay, so in the room, the microphones are on above you, so you can holler out questions. And I believe Dr. Zimmerman is online managing questions as well. So we'll just open it up to questions.
Might need to pass the lapel around. Do you happen to look at the bone also? I mean,
Yeah.
you don't have to go through all that.
Yeah.
You did.
We do quantify bone health in our animal model. We primarily rely on CT to do that, but we have looked at the articular cartilage, and their bones look terrible by 56 days after injury. We're now looking at, if we can find a way to restore muscle health, is that enough to help protect the bone? So we're also longitudinally sort of making sure that all of our muscle measures are associated with a bone measure.
So in the eccentric study, you were reporting that you didn't.
Oh, in
bone
humans.
In that compared to.
In humans, no, but that's
Okay.
part of the clinical trial that's running right now. Yep, good question.
So I guess I'm gonna step it all the way back.
Do
That's
it.
the piece where I don't see the connection.
Okay.
That is, what actually is going wrong at the point of the ACL break? I don't see why that should have any effect on the muscle,
Yes.
independent of an effect on activity. Right, so thinking about that, I can see if the ACL, you tear the ACL and I decide I'm gonna put my leg up.
Sure.
Right now I have no activity, I'm gonna have problems. But what is it about the ACL tear
Right.
that's causing?
Right. So it's not, yeah. So this is a question I commonly get from muscle physiologists. It's like, why? Right? It's not disuse, right? There is some disuse relative to normal activity, but it's not complete disuse. There's a change in the basically sensory signaling that hits the nervous system. And I think a lot of these changes early on are probably dysregulated because of the electrical activity or the contractile activity that's just not coming back to the muscle. There's also a pretty big inflammatory response. So some people would contend that when you look at the muscle, like with things like MRI or samples, like the muscle at the quadriceps muscle that closest to the knee joint looks the sickest, right? There's some cool data to say that the knee joint is kind of like a sieve. And when you load it, it sort of spews out like inflammatory cytokines that may very well travel up to the muscle. So it's complicated. Like, why exactly is that muscle so vulnerable? I don't know, but I will say in other joints that are reliant on ligaments to promote joint stability, what we're seeing here is not unique. So the ankle, the knee, that need ligaments to promote stability to the shoulder, you see this inhibition effect that starts to have this cascade of issues. The hip is not that way, right? Because it's sort of reinforced by bony structure. So it's this unique sort of paradigm that lack of ligament leads to chaos. Yeah.
Because part of why I was curious too is thinking back to the video you showed is, so prior to surgery,
Yeah.
there was pretty good muscle control still.
Yeah.
We don't know, I don't know the time between ACL tear and
A couple weeks,
surgery.
a couple of weeks.
Right, so a couple of weeks, so the muscle's still well preserved, but then the day after surgery.
So in those people,
the
yeah.
muscle tone is not prepared, is not
Yeah,
preserved.
it's
So-
very terrible then. It's also that pre-surgery, people look worse than they did prior to injury, or worse relative to their contralateral limb. So there are two things to consider. Is the surgery causing an even more problematic response? And I think from a neurological standpoint, there's kind of a lot of data to substantiate that. And, you know, would people be better being ACL deficient? Maybe so, but in those that are ACL deficient, they still have issues relative to their own contralateral limb. So they're still depressed.
In terms of, with an ACL tear,
Yep.
is there a difference between the kinds, whether you can be eccentric or concentric kinds of contractions, or is there an effect on, in other words, the destabilization of the joint? Does it affect the kinds of stretching that can occur in the muscles, so could that actually be a part of it? Is to, when you're putting weight on the joint, are you changing the way you can contract or stretch a muscle?
I don't know if can you say that again?
What I'm just thinking about is, in this last part where you're talking about the eccentric and the, the effects of that on muscle. And I'm just wondering is if I blow out an ACL, do I stress my muscles differently? Maybe going down the stairs, but maybe going up the stairs, I can still stress them the same way.
I've never.
So am I exercising it differently because of the loss of the ACL?
I don't have data to directly speak to that. So is the instability of the knee causing a different mechanical stimulus when eccentric exercise is applied? That's what you're saying? I don't know. Maybe. I guess that we would, some of Ryan's clinical trial might get at that with like controls. That's a good question. I haven't thought about it that way before. I'm not sure to what extent that knee instability really changes the stress at the muscle level. I don't know. Yeah, that's a good question. Yeah.
So why the quads, not the hamstrings?
Yeah, people have looked at that quite a bit. It seems the quads are like much more vulnerable, even when there's an ankle injury. What shuts down soleus and like quads? So it's just a very vulnerable muscle group, much more so than the hamstrings. Now, there is data to show that the hamstrings aren't doing well, but comparative to the quads, it just really stinks. We don't really have good data on what happens after and what is it about the trigger of an ACL injury and why is the muscle group, this particular muscle group, more problematic than anybody else? We don't know.
So my other thought is it seems to be there is some sensory connection, I think, between the quads and the ACL.
That's true.
Disrupted during injury, and it's not replaced with the reconstruction. But if you look at the Bear procedure, which is repairing it, are those folks recovering their quads better?
So the Bear procedure, for those of you that aren't familiar, is basically putting like a sponge in the middle of the knee joint with like growth hormones to sort of regrow the remnants of the ACL back together. Some work that's being done in Boston. What I will say is that when people have looked at the histology of an ACL graft, it's free nerve endings that repopulate it. So I don't know what is going to be repopulating these like remnants, or if that isn't quite the case in these Bear procedures. But it's confusing because the quad also really shuts down when there's an ankle injury and there isn't that direct ligament connection that you're talking about. So maybe that helps to partially explain the difference between the quad and the hamstring, but it doesn't explain ankle to quad. So it's.
You don't get atrophy though with an ankle injury to the quad, like you do with an ACL.
You don't get atrophy. Well, correct. There's other things that are substantiating the atrophy in ACL injury, and the extent of the inhibition is much greater than with an ankle injury, but it's still a vulnerable muscle, right? So
It's tricky.
it's tricky. In part, that's why like some preclinical work is probably needed because this is a very confusing paradigm.
So the other thought I had about so when you're talking about eccentrics, which is true, so for creating hypertrophy, right? Strengthening at a lengthened position is the most beneficial to get the most hypertrophy. So going down to end range, getting in a very lengthened position and loading that lengthened position. So, which you kind of show, right? So the eccentrics in as far lengthened as you can get is the most beneficial. The problem is early on, you can't get there, right? So after ACL surgery, how many weeks out are you before you have full?
So our
flexion?
people that had long-lasting benefits were working in mid-range,
Right,
right? So that, even if you're not completely mechanically stretching the muscle out,
okay,
you still get benefit from eccentric
Yeah.
exercise because these people were very mid-range. We avoided, full extension and full flexion. So yeah, there's some compelling hamstring studies to show what you're talking about now, that really long lengths, but it
right.
seems to be, you don't quite need that degree of mechanical stimulus to make it work.
Gotcha.
Yeah.
Thanks. So I'm curious about some of like the neural stuff with this as well. Is there like a really, really cute TMS coil that's been invented to look at like excitability or any histological changes with that?
Like at the
In
rodent,
conjunction with the mouse model that you've done.
a very small TMS coil. Well, there's
I doubt
ways.
it exists, but
Sure, there's ways to stimulate the brain,
Mhm.
like directly in an animal model. That's done all the time.
Mhm.
I have not ventured there, but I suspect at some point that this would provide an opportunity for that and we could take a look. Yeah.
Just going back to what you were talking about with the long-lasting benefit of the eccentrics, can you define what those benefits were, like how you quantify those things?
Yep, so we looked at isometric force production, we looked at their volitional activity during that production, and then we looked at their knee biomechanics. So they had improvement in strength and volitional activation, they had greater knee flexion angles, and they had more knee extension moments, and this was at somewhere around 9 months. Perry Gerber's done some work and shown that it holds up with MRI for 2+ years. So it appears to be pretty long lasting. Yeah.
So, in a lot of the patellar tendinopathy models, there's kind of been a shift from eccentrics to heavy slow resistive training for the progressive loading aspect. Do you think progressive loading could be a therapeutic form
Yeah, muscle is very lazy. And it's very easy to swing into like a muscle breakdown situation. So I think just in general, like you have to stimulate it, whether that's with e-stim or whether that's with like mechanical or progressive overloading. It's not going to respond to like cute little exercises, generally speaking. Like you really kind of have to ramp it up a little bit for it to be responsive in this situation. So maybe, yeah, it would be beneficial.
2 questions about your model. Is the rate that you tear the ligament bear any resemblance to the rate at which an athletic ACL tear occurs?
I have no idea. I know that it works. I know that it creates a similar amount of force in our male and female rats because we take a look at that. So I know that we have a pretty consistent injury, but I have no idea, like to what extent that maps on.
Just wondering what additional tissue capsular ramifications there may be if it's a different rate.
Yeah.
And then regarding some of those intra-tissue findings that you looked at in your model or that you spoke to, I think you guys looked at it, right? The NCAM.
Oh, yeah.
The glue. Have you looked at any of the rats that you would go back and maybe do a reconstruction and see what a second violation of the knee joint might do to that?
So that in part is why we've stuck in a rat's like the size of a water bottle. A mouse is like the size of a mouse. A computer mouse, maybe a little smaller, but so that's why we've stuck at this level because it provides an opportunity to do reconstruction. Now, what we're doing right now are interventions where they're deficient, and if we find a way to restore muscle health, but that does not help the knee joint, then I think that necessitates the surgery, right? And or some of these other questions may necessitate the surgery. But right now, we're sort of looking at this paradigm of like muscle health first and see what subsequently follows. But it may be that both things are necessary. Yeah.
I have a question.
Your PhD student doing the EMG work,
Yeah.
it looked like you presented some data on male versus female rats.
Mhm.
And I don't know if those were looked at. It looked like there might be some differences just visually looking at your graphs.
Yeah.
Do those sex-based differences replicate the sex-based differences that we see in people? Or are they, do you think you would, are they valid? Do you think they would hold
Yeah.
in a human population?
Yeah, so we've come to understand that males and females do not recover the same. And that's also true at our preclinical level. Our males have more of an atrophy response than our females do at the preclinical level, which doesn't necessarily map onto the clinical condition. Some of our hypotheses around that may be like the protective nature of hormones and its ability to handle reactive oxygen species in a female rat that might be protecting the muscle in our animal model a little bit better than our males. And so that's sort of our line of thinking right now. But they're different. So what we're doing at our level is we're casting a wide net and we're collecting it all. But I would say our male rats are more representative in general of the condition that happens in the clinic.
Are there any questions online?
No, there are none online, Liz, that I can see. Any other in the audience?
I don't think we have any here.
Okay, well, I can see why you were so excited to have her come, Liz. This has been great. Thank you, Dr. Lepley, very much. We really appreciate this. It was a great presentation and I learned a lot.
Good.
All right,
Cool.
thanks.
That's that.
Thank you very much.